So there is this huge work called, that have been called Marine Ecosystem Assessment for the Southern Ocean. It's a huge collaborative work that has been initiated in 2018 with more than 200 scientists in 19 countries that came up with the idea to make a big review of the status and change of Southern Ocean ecosystems. It's 24 research articles that has been published in a special topic in Frontiers in Ecology and Evolution. And I came with this idea that it would be great for the summer school to make a little review of these 24 articles. And it's actually hugely dense. I really invite you to read these papers because it's a lot of knowledge and wonderful summaries of all the studies that have been done and the drivers. And actually last year came out the summary for policymakers of this work. And actually today I will attempt to make a little summary, but I will not even cover, I would say, 1% of what has been done in this work. But I hope I give you a brief overview that give you the one to go a little bit more deeper, or just have like a basic knowledge on what is going on in terms of this ecosystem. So there are a lot of texts because
Sorry to interrupt. I just thought about something. Sorry to bother you with forwarding and interrupt you, Sarah, but just with the list, if you can check, even if you don't change the time, if you can check that you check the right something, that's okay, however. So I know that everyone seen it. Very sorry.
no worries. Do you have any other announcement coming in? So I'm ready. So yeah, there are a lot of text, but I think, this presentation, I will then give it to you, and you have lots of reference. And I think, to keep and have an idea, I could have also just talked through it, and then you won't have it printed and the reference to the source. So it might not be super fun always, but it is the reason. And also because I don't know all of that. I will first ask you a question. So what do you think now from the presentation you had from Elizabeth, myself, and like your knowledge? What do you think are the main drivers for a marine ecosystem? Like, can you point some drivers? Physical, anthropogenic, biological drivers. Wake up! Drivers of change of marine ecosystems, Southern Ocean marine ecosystems. What could be the drivers?
Simply environmental drivers like temperature, salinity, like that.
Yeah. So change in temperature, change in salinity, sea ice, food, nutrients. Sorry? Fishing. That's all? Okay. Winds change, yeah. Pollution. Acidification. There is something you didn't mention. Sorry? Yeah, ice shelf melting. Ice shelf calving. What else? Okay, I think we kind of cover basic. So here is the a list of all different drivers of change in ocean habitat and ecosystems. And I'll just go through it quickly because you know that better than I do actually. But you have different atmospheric and ocean drivers. You have what they call also northern drivers, which mean like, I would say climate variability index or oscillation, plastic pollution, range shift. It can imply a lot of different things. But ice shelf collapse, so it will have an impact by altering stability of the water column and the mixed layer depths. Sea ice loss, we talk a lot about it, but increased vertical mixing, nutrient supply, it will alter these processes. Glacial retreats that will have an impact on primary productivity, pelagic and benthic food webs. Ocean acidification through the ecosystem structure function, carbon export, ocean warming. I will go into that later. Shallowing of the deep water that will have impact on ice shelf sheet instability, benthic habitats, and change to primary productivity. So, for example, how change in ice sheet or ice sheet calving will have influence on, for example, benthic ecosystems.
How?
Yeah, you're the specialist. I'm looking at you.
When the ice melts, brings great input of organic material to the water column, so net shifting primary production, and then all this organic matter settles down and provides food for the benthic organisms, so maybe change the function of the.
Yeah, but there are also two factors that we never really think about. One is like a physical mechanic factor. That would be the movement of this iceberg on the seafloor are also deteriorating a lot of benthic ecosystem, and some that are short-lived will settle again and be able to develop, and one that are long-lived and that are like build up like strong communities, they will much more struggle to settle back and recover. So it was one thing. And it's also a change. I mean, if you see these ice shelves on top of the benthic ecosystem, it's also changed the light, just simply. And it will change all the light penetrating under ice shelf inputs it could have. So just some example that one drivers could have multiple consequences, and it will really depend on species and the type of habitat and ecosystem in general. So I will start with primary production. But we will see primary production, zooplankton, fish and squids, birds and marine mammal. And some I will go faster than others. So you have different type of primary production. So you have the age phytoplankton bloom, the mixed layer phytoplankton blooms, deep chlorophyll maximum blooms, phytoplankton communities as well, phytoplankton beneath the sea ice. And what they did is they review 3 compartments of phytoplankton, sea ice algae, so phytoplankton in the water column, sea ice algae, and at the deep chlorophyll maximum. And each time, they made an assessment of the status, whether it's going increasing or decreasing in the past and in the future. So we'll start first with the phytoplankton in the water column. And based on satellite observation, it suggests that phytoplankton biomass in the mixed layer, so this compartment, has increased over the last 20 years in most, but not all, parts of the Southern Ocean. Something also I wanted to highlight is that I'm making some general statement of Southern Ocean as a whole, but there are very different regions in the Southern Ocean that have very contrasting trends. Saying that because, for example, for emperor penguins, we are observing like very different things depending on where we are in Antarctica. So for ecosystem, never make us, we can have a general picture and trend, but there are also a lot of variability from one region to another. So basically in the past it has been increasing, on the contrary, the primary production at the base of the mixed layer depths has likely decreased over the same period, so here at the deep chlorophyll maximum. And for the future, we don't know. Yeah?
Do you have any idea of the mechanisms that are advanced to explain this change?
For this? Or this?
I was not aware. Do you know why this is?
There are lots of different hypotheses. Maybe Elizabeth is. Do you want to? Yeah. And also, the recent also sea ice changes as well is having a positive effect actually on primary production in the surface. Within the mixed layer, because it means like more surface covered by sunlight and more nutrient coming from sea ice melting or ice shelf melting, and more stabilization as well, because not sea ice forming less. So more stability in the water column, but at the same time we don't know in which, I will talk about that data just after. We don't know then the future, because there are a lot of negative feedback to that, and in the long term it might not have this effect, but we don't know yet. Yeah, so some of the future things that they are saying might actually be wrong in maybe 10 years, based on the knowledge they will get. So never think it's a static system, even these big reviews. So yeah, primary production in the Southern Ocean will likely increase over the next 100 years. So with more primary production in water column and less in sea ice. Low confidence in this projection, it's actually what I'm talking about, because there is a difficulty in mapping the supply mechanism of key nutrients, so silicate and iron, and also understanding the effect of multiple stressors, irradiance, nutrients, temperature, PCO2, pH grazing, on different species of Antarctic phytoplankton. Also, one driver will not have the same influence on each species of phytoplankton. So the distinctive nature of microbial community, autotrophic community, so the dominant of species, the phenology, which means like seasonality and their ability to adapt to this seasonality, spatial distribution of primary production will likely reduce with shift to our community more dominated by small and flagellate species. So Elizabeth talked about that already this morning. And endemic Southern Ocean phytoplankton species are losing out to temperate species, actually. But there is year-to-year variability in primary production in the Southern Ocean that will also likely increase with the increasing prevalence of marine heat waves, another component as well that is like more like an extreme event coming more frequently, but that will have also an influence. And the example actually that will be relevant to Martin is that the environmental changes driving this complex biological response are complex and not well understood. So for example, you observe a shoaling of the mixed layer depths due to decreased vertical mixing. So for example, you form less ice, you have less mixing, you have more stability of your water column, but simultaneously you increase the mixed layer irradiance, you increase the grazing, you increase the damage, ultraviolet B damage to the cells, and you decrease also the nutrient supply because you don't have this mixing, and also the living space for certain species. So you have like counter effect balance, and we don't know in the long term which effect will dominate or not, and how this will actually be true in the future. Okay. And this is for sea ice algae, so it's forecasted to decrease because sea ice algae will be affected by reducing in the sea ice extent and concentration. And also this change in sea ice are way more spatially heterogeneous and non-linear than for, I would say, basic phytoplankton, for water column phytoplankton. Because you see the change in Antarctic sea ice are very different with region where, until recently we saw some increase in sea ice trend and some region where you have like a huge decrease. And the extreme, I'm not sure I'm right to say, the extreme event we have seen in the Antarctic sea ice extent in the last 7 years also, might affect how all these links are made. So, this is not good for the ecosystem because, as I told you yesterday, sea ice algae are a crucial bridge between the period of low and high productivity. For many trophic levels, the sea ice algae is really the little food that remain during this winter time when there is not a lot of thing around, and lots of species are dependent on it. So if this disappears, it has some consequence as well on the marine ecosystem. Okay, I will just make a zoom before talking about zooplankton on actually Antarctic sea ice systems that I talked yesterday about. So you remember this diagram that we saw yesterday. I hope you remember the species that dominate in this kind of system. Maybe not. Oops. So, brief reminder. So the magnitude and timing of the spring ice-associated algae bloom is directly related to sea ice extent and duration of the previous winter. So that is important to know. And so ice algae is a food source for larval Antarctic krill during late winter and early spring. And there is complex under and intra-ice habitat in providing shelter for larval krill. And there is also an importance, and I didn't talk too much about that yesterday, of the sea ice drift pattern in transporting krill larvae to area in which they can survive and develop in spring. And there is a strong relationship also between sea ice condition, influence on ice, algal, carbon productivity and export, and subsequently on the benthic community within this sea ice environment. So within this context, if we observe some sea ice changes, and we do observe in some regions some sea ice changes, it's possible that copepod can continue to survive. So copepod are mainly within these brine channels, and you have like 4 different species among these 2 groups that dominate. So in thinner and less frequent ice cover, under different scenario, the plasticity of their diet and their life cycle might actually be key for their continual success. So copepod will be doing great. But krill not much, because what I told you just before and yesterday. And still uncertainty about that. There is an important spatial segregation between krill and salp population, but they show that their feeding behavior actually, they have similar diets. And that might also enhance competition within the 2 species. So if sea ice tends to be less, and you have like copepods thriving in like warmer waters and less sea ice, and then they have the same kind of diets, then there will be like competition appearing. So I'm just introducing that not only physical drivers, but now you have also competition drivers that will interplay because of a climate change first scenario bringing it to here. And then temperature, sea ice, and chlorophyll A drive large-scale population segregation between these 2 species, and that has been seen mostly in the Indian sector of the Southern Ocean. But actually, if there is change in this temperature and sea ice and chlorophyll pattern, actually this like separation between these 2 species might not be so clear. So observation from the West Antarctic Peninsula indicate oscillation between years of salp dominance and years of krill dominance. And this is something very interesting to investigate further, what is dominating each cycle. And the rapid reproduction capacity of salps under favorable condition could change current ecosystem structure and alter behavior and distribution of krill. So salp is very different in terms of what for as a food source. What is the difference between krill and salps? Alors, sorry? I mean, before that, so there is salps are gelatinous. Not before that, but very simple. They are gelatinous organisms, and so in terms of nutrition quality, it's very different. And also, we don't know the implication of eating salps instead of krill, because they might have not the same quality for species. I don't know actually about the lipids. Good question. Yeah, it's quality, but I don't know how much content and how they differ, and yeah.
No, I don't know.
Also, and sometimes it's like you pull your hair out when you work on animals and try to understand what is happening underneath ecosystem. But the different taxa do not respond in a consistent direction, positive or negative, across all environmental perturbation. And that highlights the complexity of networks of interaction between the physical and biological variables. So yeah, it won't be going in the same thing. And for example, increasing storminess, which is one of the potential driver, will produce positive response in the pelagic nutrients pools, possibly due to deeper ocean mixing, bringing nutrients to the surface. But at the same time, because they suggested that we'll have increase in storminess, but also warming temperature, and the warming temperature might lead to increased stratification, trapping nutrients at depth. And so then it's between all these factors, you will see which one is dominating and how it's influenced these species compared to these species. And it makes the complexity to state on the population status. So also, the fact that, for example, the dependency of silverfish on platelet ice for their early deployments is something that could be critical, because if there is a change in seasonal ice cover, it will be very detrimental because they're really dependent on this perennial multi-year ice. And highly adapted species, so such kind of species that have their life cycle totally tied to a type of ice environment, will also be difficult for them. Okay, zooplankton. So this table is summarizing the main effect of different physical, chemical drivers here. And you have different group of zooplanktons. And so mean and known are the influence is too different between species that we can't answer. Low influence in blue, in red, high influence, and medium influence. And I will go through different groups. So Antarctic krill, we already talked about it yesterday, but I highlight sea ice, temperature, ocean acidification. And yesterday I didn't fully reply to one comment of one of you to the acidification. So I will today, but first temperature. So here is a diagram of the different compartment of the activity of krill. So ingestion, assimilation, reserves, metabolism, egestion, excretion. And if you increase the temperature, it will have an influence on the metabolism. And so adult krill has an optimal range between 0 and 3 degrees. And they are able to grow and reproduce if the metabolic demand is met. So that means they will have enough food to have their metabolism working. But their performance actually, performance which mean like ingestion, assimilation, excretion, exhaustion, will likely decrease if temperature goes above 3 degrees due to thermal stress. So that means they will need more energy to do all of these things, so more food, and they might not be able to get this more food and to do all of that. And larval and juvenile krills that have lower energy reserve compared to adult will be more affected, of course, because they will run out of energy earlier. Also, temperature above 3 degrees will also negatively impact the embryo development, causing rapid decline in egg hatching rate and increasing malformation. So that is just a consequence of temperature isolated, without thinking even of change in sea ice. So yesterday I presented this paper. So there is a link between regional warming in the western Antarctic Peninsula, contraction of the krill and reduction in the krill lead to change in sea ice and diatom loss, and all the three together linked. Ocean acidification. So ocean acidification. So it's still unclear, actually. I looked at it because I was not replying really precisely to your question. But embryonic development and hatching are negatively affected by change in PCO2. Okay, but at the same time, adults actually are, for now, quite resilient to this level of PCO2 above 1,000 micro ATM. And actually, they are able to maintain an acid balance in their body fluid. So the only study combining impact of acidification and increased temperature suggests shorter-term effects were primarily pH driving, whereas long-term effects on growth and survival were strongly driving by temperature, but no effect of pH. So more study coming, but we know that there is an effect on the embryogenic development and hatching. Okay, so I won't go towards the other ophiocid because it's quite similar to krill in terms of the drivers, but I will go towards copepod. So copepod, unknown. Voilà. No, I'm not going to say something. Copepod will do good, because, we don't know. It's still unknown because there are different species. But there have been studies that made some huge effort to standardize sampling methods, because if you want to compare what happened like 70 years or 80 years ago, so they have been having some sampling that was way different from now. And it's actually, this graph is like sea surface temperature for different species of copepod. And the difference is like 70, so Discovery, it was a campaign like 70, 80 years ago. And contemporary is, now the time of the paper. And basically, they show that the biomass of dominant copepod, there were 2 studies on this campaign. Sorry. The biomass of dominant copepod species have increased in abundance in the last 70 years by 20 to 50%. And also that in this sector, so South-West Atlantic sector, there is, they kept their same spatial distribution despite there were a mean increase of temperature of 0.74 degree. But they didn't change anything, they just stayed there. And so they were inhabiting like warmer area without any problems. So they were doing great for that. But I think the reason it's all because I think there might be some influence that is different between species or sector of the Southern Ocean. So they can't conclude in a global scale. Then, the SARPs. I already talked about the SARPs with the krill, but we'll go a little bit more in details. So, height influence, so it doesn't mean it's positive or negative here, it just means it has a high influence. So, sea surface temperature, ocean circulation, and extreme climatic-related event and climate indices. So, this species, sorry, it's not in italic, Salpa thomsoni, which is like the main dominant species of SARPs in the Southern Ocean. Its distribution strongly correlates negatively with sea ice cover and positively with the warm water intrusion into Antarctic region. And a reanalysis of multi-year data set on SARPs postulated that temperatures strongly drive the SARPs population, this SARPs population dynamic. And so observation from 1926 to 2003 about circumpolar SARP data south of the southern boundary show actually that the SARP increase over twofold. You see all the blue part in different places close to the Antarctic continent. So it suggests it has a general increase in density and a circum-Antarctic southward expansion in their distribution, which mean they have shifted significantly southward and intruding into area generally occupied by krill. So it doesn't sound very good for krill. But this species, as krill, is extremely patchy and they have large interannual variability, so we don't know yet a lot of things about this species. Yeah, and the rest is just anecdotal. So, wait, before I show you that. So, how does the SARPs versus the krill will have an effect on carbon export? Like, what do you expect? Is it the same, different? How it will change? And this will influence maybe some also biotic questions, but it will more be of the interest of Elizabeth, I guess. Do you have an idea? Same, more, less? No, you're on a good direction. So SARPs, they produce 4 times more fecal pellet than krill. And this fecal pellet contributes equally to the carbon flux at 300 meters. But krill fecal pellet are exported at 72% to 300 meters here. Boop. But for the SARP, the 80% of the SARP fecal pellet remain within the mixed layer and doesn't go all the way to these 300 meters. So that means that, I mean, it's only one paper, and I think there are contradictions as well in that, but it's one of the most recent papers I found on this. The declining krill abundance could lead to decreased carbon flux as well. And I mean, this was based on the peninsula again, but that means the peninsula may be a less, I think, of CO2 than it used to be due to that. Yeah. Okay, pteropods. So I talk about it because they are a little bit present as well in the Antarctic ecosystem. Pteropods have like carbonate calcium shell. So of course, they will be a lot influenced by ocean acidification, but also by temperature. So the overall pattern is dominated by an alternation between height and low abundance of the pteropod that is negatively correlated with the ENSO index and positively correlated with SST, CIS, and primary production. And that's what we know, and I think, this species, of course, will be impacted in the same way, in a similar way as krill. Okay. So now, Southern Ocean fish and squid. Look at the time. Yeah. So here, again, you have blue, no data, negative impact, mixed impact, positive impact. And they kind of gather the drivers into climate change, pollution, CS loss, fisheries. They have a lot of information from fishery data, but not much, either it's not easy to sample all the species all the time. So here you have like the distribution, the historical impact, the driver of historical impact, future impact, and yeah, the driver are here. So now I will just talk about fish in general, so considering all these fish. So, from a lot of studies, fishes are expected to be affected by increased temperature, of course, and their special adaptation to colder temperature are actually like the protein, the anti-freezing protein will not help. A recent study predicted that most species will undergo a major poleward as well, shift in their distribution. I guess all species will just climb on the Antarctica shelf soon. Yeah, and then this study actually was looking at multimodal ensemble mean of the predicted change in latitudinal distribution for 10 species and for 2006 to 2055, and same 2050 to 2100 almost. And actually they show this major poleward depending on, even more in a worse scenario, I would say, of this fish, lanternfish species. They reported as well a change in body size with smaller size myctophid and life stages going more and more south, possibly displacing, so it will be competition again, those that are in this region originally. And ocean temperature as well will change the vertical distribution and behavior of these fish. And this has high consequence as well in terms of where they go to feed and where they aggregate and how much energy they expend to go to these different depths, given their usual habitat. Antarctic squid. So it has not been fished, not still. And actually it's the only one, it's like the salps / copepod squid that is will be okay to survive in the future. So maybe we'll eat more squid instead of krill. I mean, we don't eat krill yet, but soon. And so a recent long-term study showed that the habitat of different species of squid have changed over the last 50 years despite their trophic levels remain the same. So that means they can change habitat without having too much problems to find food. And this graph shows from 1976 to 2016 for different species of squid, the delta carbon 13, sorry. And this gives you the change in the habitat. So if the delta going up, it means a northward shift. And if it's going down, it means like a southward shift. So basically, these species have changed, these species didn't change, and these species also have changed, but not in the same direction, and they all are stable and being able to feed and grow. And this is another study, so it's not only Southern Ocean, I think it's all squid everywhere. Demersal, which mean, what mean demersal? Maybe I didn't explain. Demersal that is on the bottom, that usually live on the bottom. Bentopelagic, that mean that they will oscillate between the water column and the bentos, I mean in the demersal way, I would say. Pelagic, they're always in the water column. So basically they show that from 1960 to 2010, they increase in terms of abundance. It's a standardized abundance index in any type of compartment of the water column. So why? I mean, it's white and there is a unique set of biological traits, including rapid growth, short lifespan, life history plasticity, which may allow them to adapt to change in their environment. And actually, the increasing temperature will accelerate their life cycle because they have high thermal ranges. And actually, the temperature will improve their reproductive success. And also, they are not fished. And their predators are fished heavily, like, for example, the Antarctic toothfish. So that means they increase and their predators decrease. So all going well for them. Pollution in general effect on Southern Ocean fish and squid. So there is not a lot of study actually that have been able to investigate the impact of pollutants on early life stage of Antarctic fish. They think there is a growing evidence that environmental pollutants, which could be polychlorinated biphenyls, polycyclic aromatic hydrocarbon, dioxin, have adverse effect again on the embryonic and early larval development in fish. And also this sensitivity could be enhanced by stressors, other stressors. So imagine you have like a fish that already struggling with change in sea ice, temperature, and then on top of that you had pollutants. They might be more sensitive to pollutant than when they are not like struggling already to find food or habitat to develop, rest, hatch, spawn, sorry. And so this is always to consider, like it's not only one factor, is altogether what is the effect on the species. And for example, in Antarctic dragonfish, combined to exposure to ocean acidification, warming, there is alteration in metabolism and developmental rate in embryogenic stage. And so if you add like pollutant pressure on top of that, it's not good. They also said that the mercury is also really bad, but they don't have like yet some studies to, I mean, only on some certain species, but it has like a lot of impact. Yeah. Yeah, and then the extent of this impact may have population level effect in the future, but we don't know yet about that. Okay, so yeah, so this is the Baltic communities. This before last compartment, and it's what we were talking at the very beginning. So you have like the undisturbed scenario here, but then you have like the different drivers and how it will affect them. So ocean temperature, we talk a lot about that, but for example, you have species that have a really thermal range that is, they can't sustain increasing temperature basically. But you have also the problem of invasive species in the benthos, and for example, king crab abundance increasing and increasing and increasing, and that will also replace some other species in some temperature range. Decrease in sea ice, that means there will be more opportunistic and fewer specialized suspension feeders that could have an effect, and I will add to that maybe change also in the light coming in on the benthos. Increase in iceberg scoring, so the more fast growers will be more selected, and the slow the slow growers will be going down. Ocean acidification, because there are lots of calcifying corals and other species. And fishery pressure, of course, because most of the fishery will score as well the floor. Yeah, so this is what I already said. So the sessile benthic species, for example, the reef builders, they cannot escape, and so under unfavorable condition, they will just die, I mean, and not be able to change. And same for species with argonautic shells, they will not be able to survive in ocean acidification. Okay, so the last part, so it's birds and marine mammals, and what time is it? Yeah. I didn't make some case for each of them because you can clearly see in red is the negative impact. So you have like, again, all the drivers. So from one compartment to another, it changed how they presented. So oceanographic processes, CS processes, fishery interaction, global pollution, health, and disease. That can also is important in birds and mammals because they travel so far within the Southern Ocean that they can be contaminated by different things from our actually inhabited continents. Did you heard about the avian influenza? Yeah, and the consequence. Yeah. Who don't know about it? Martin. But basically, it's a virus that has been propagated. That started in South America, and as the birds are, for example, Southern Ocean birds are flying between the subantarctic islands and the Antarctic continent, and Southern America, it's spreaded, and this is very dangerous because it could decimate some population very quickly. And actually, this virus originated from the wild, but was not leading to the death of individuals. And then it was transmitted to industrial farming, and then in industrial farming it mutates and then gets actually deadly. And then it transmitted again from farming to wild animals, and then having this huge consequence. So thank you, industrial farming. As you can see here, for these different groups, it's a lot of reds. Warmer temperature, extreme weather event, change in sea ice, fishery that remove the resource of the animals, bycatch as well, it's a main problem, pollution, human disturbance in some region, for example, in the peninsula where there is a lot of tourism, and all of that have like a very important consequence on top predator, especially that they don't reproduce that fast, and they are long-lived species. Yeah. Where do you see that? Here? On the depredation? No, not fishing. No. It's so fishing leads to bycatch, and but it's as a mixed effect because they feed on fisheries. So for example, sperm whales or killer whales, they will fish on this long line toothfish fishery. I mean, they will eat on this long line toothfish fishery. And actually, they've seen that there are some groups of, subpopulation that actually specialize into fishery in the same way that will specialize in for example, killer whales, they specialize in a type of prey and then they stick to it. So if they will eat, there is subpopulation that will eat only elephant seal, perhaps there are subpopulation that will eat only a certain type of fish, and there are subpopulation now that eat only long line toothfish. Long-line fisheries toothfish, and so it has mixed effect because it sustains some population in some way, but also like it has an effect of bycatch and negative effect. So that's the reason I think they put it mixed. Yeah, is that clear? Yeah. To finish, I will link to that. I wanted to show you one study that we've been doing on emperor penguins, trying to understand whether they will survive or not to change in sea ice. And I will go back to that later. I think I will start from here. Yeah, there are different studies where we investigate actually the link between sea ice and emperor penguins, and you will see at the end where I'm getting. But we started in the most study colony of emperor penguin, which is in the French Antarctic Station, Dumont-D'Urville, where they started to follow this colony in 1979 until now. And it means like every year they count the number of eggs, and the number of eggs that survive, and the number of chicks, and until they know that the breeding success. And actually, we were wondering the effect of change in fast ice dynamic to the emperor penguin colony, because when they breed, they actually need to go to open water access to feed and bring back food for the chicks. And we were wondering whether these like small polynyas, very small polynyas or water opening were having an effect, and how it changed from one year to another. And actually, so this is the time series of reproductive success. So you have like different parameters, and I won't go into the detail, but like fledging success, hatching success, and breeding success from 1979 to 2017. And basically we looked, so we went through historical fast ice images where we calculated the distance to the nearest open water access in kilometers. And so this is the nearest land fast ice edge in kilometers. And the color corresponds to the different months, but we don't really see with this. But if you look at the pink square here, it's each time where you have like almost no breeding success, almost zero total breeding failure. And this corresponded each time that the sea ice was very wide, so the extent was very important. And actually in emperor penguin, if you have no sea ice, it's not good because they need a platform to breed and have all this chick from May to October. And also because the sea ice sustains a whole ecosystem where they may be able to feed on it. But if you have too much sea ice, it also has an impact because they don't have time to go all the way to feed and come back. And so that leads to the death of all the penguins. And we saw this relationship between breeding success and the distance to the nearest land fast ice age. That was quite important. And then, just not, yeah. And then we were wondering, okay, so what is the best place to live in Antarctica in terms of fast ice? And because we always say they need fast ice, okay, but you see that some colonies, the extent of fast ice will have very important consequence on the population dynamic, and other colonies not. And we were like, okay, how can we understand sea ice changes if we don't know actually what kind of sea ice they use for their breeding cycle? With actually a colleague at IMAS, Alexander Feder, we pull lots of different variables of fast ice, and I'm not sure I will go in. I'm not going to go in the detail of this fast ice dynamic variables, but it's basically the amplitude of the fast ice, how much it breaks and forms, what we call the volatility, the maximum, minimum extent of fast ice. Different variables associated with the dynamic, and we tried to understand whether there've been some patterns around Antarctica and whether they were using exactly the same fast ice to breed. And what we found, so basically we looked at different parameters, so I just summarize this, and we made some cluster of these parameters, and then we plotted this cluster to see where they were actually. And what we found some very different parameters depending of where you are around Antarctica. And there are places where you have kind of some parameters that are all together, and some places where it's like a little bit more mixed. So you can see, for example, in the Ross Sea, you have all this cluster 2, the black one. In East Antarctic, it's dominated by greens, but you have also some other type of habitat. This place is dominated by these 3 colonies have blue type of habitat, and then the Weddell Sea part is all red. And actually the black contour that you see is the genetic metapopulation of emperor penguins that we know, and so we know only 4 of them. And actually we found, I mean, they only sample in this different area, and they found some genetic metapopulation. And we found that there were actually some correlation with this metapopulation. And we think that actually some population might be more able to adapt to certain fast ice conditions than others. And actually this is super important because we always consider emperor penguin as a wall circumpolarly in terms of their habitat and in terms of change of sea ice, but actually they might not be affected by the same sea ice conditions. So under different scenario, some population here will not be affected in the same way as here or here or here. And in terms of conservation, it's very important. And I'll finish on that study, but how do you know if emperor penguin are like increasing or decreasing? Like most of the emperor penguin colony around Antarctica are inaccessible. You can't go by boat, you can't go by plane, you can't go with anything. So how do you do that? So an idea from one colleague was to use satellite data to count penguin from space. And it's a huge work. So basically you look at all these satellite images and you visually try to find some guano spot. And from plane it looks like that. And we did a study where we tried to compare IRL count with satellite count, and then we used satellite count over 10 years to try to estimate the change in emperor penguin colony. The problem is that these penguins, when you see them from space and you count pixels of penguins, when it's warm they spread, and when it's cold they go very close to each other. And so if you take a picture, a satellite image in September 8, and then you take a picture in October 2, and it's not the same wind or temperature condition, you can have lots of bias counting these penguins. So basically, yeah, we went through all this image, and we define, okay, this is guano, this is ice, this is rock, this is penguins. And then with automatic classification, you can get a number of pixels of penguins. I mean, you have like lots of hours of correction of this classification manually. And yeah, you end up with this kind of pixels. And then you end up to these 2Ds that took ages to do. And basically, for over 10 years, we tried to, at each colony, so there are like 55 to 65 colonies, depending of if you consider if they are gone or every year. And basically, the idea was to kind of make some trends, and it's very difficult, lots of bias, and there are also some modeling simulation to correct the bias to this estimate. But what we found is that there is an 81% probability that there were fewer adult emperor penguin in 2018 than 2009. So this is from observation, but then there is another study we did where we project demographic rate of emperor penguins and under a different scenario. And Stéphanie Genevrier, with who I was doing a postdoc with, actually we wanted to convince people that this 1.5 degree is very important for penguins. And actually, I mean, from the simulation, and there are lots of bias to this analysis, especially if you come from observation like me. I'm like, whoa, how do you, like, because you simulate change in sea ice over 500 kilometers square, when you see that the emperor penguin are influenced by 1 kilometer type of parameters at sea. So, of course, there are lots of limits, I would say, to it. But what we found is that if we continue on a 2.6 degree path, 98% of the emperor penguin colony will be gone by 2100. So that was not very nice result, but the nice result was like if we were sticking to a Paris scenario, we would have still emperor penguin colonies by 2100, because they are so sensitive of change in sea ice. And what I was explaining to Dirk is they can't climb the ice shelf to go somewhere else. Like maybe they will become alpinism, but not yet. Actually, I saw some penguins climbing little hills where they are big, but I don't think they can climb the ice shelf to breed and go back and forth each time. So, yeah. And I'm finished now, but I just want to go to a last one tiny thing. Two texts from two people. Is it progress to define material growth as the only vector for the development of a community? It just in a way, the society that we build is made on this progress idea, and that leads to change in species and ecosystems, and as well impacting humans population, not only non-humans living things. Yeah, this sentence made me think for a long time how we can be progressing and growing and developing as a community without this material growth that kind of distract a lot of different things. And this is a text from, this was from Jane Goodall, and this is a text from Gilles Boeuf, which is a professor at university in Paris, and also working at the National Museum of Natural History. Saying that the human body, I will just kind of read and paraphrase it, but the human body is home of at least 10 times more bacteria than human cells. So we are made of lots of bacteria. So that means non-human living things. Every time we eat, drink, move, walk, swim, or inhale, we are invaded by these microorganisms, both beneficial and sometimes pathogenic, only sometimes. If we're made of atoms, water, and bacteria, why should we continue to display this arrogance that would isolate us from nature and animals? And how we made of it, and we always dominate it in a certain way. Humans have a profoundly animal physiology, like them, like animals, we breathe, eat, have a similar metabolism and similar behavior. We are coming from a long, bushy evolution of a group of primates among the mammals from which we separated a few million years ago. We are in a way we are not at all in a way an evolutionary culmination at the top of the hierarchy of living things. Because we always represent ourselves at the top of evolution, but if you look at really this bushy chain of evolution, we're not at all at the top, and there is no hierarchy in living things, actually, if you look at just the basic trees, living tree. So we do not need to insist on this peremptory distinction with regard to these other animals. The more we study them, the more we know about their biology and behavior, the more we learn to discover them, to consider them, and to rethink our place and their place in this world, as well as our duty towards them. Gandhi said that the development of a civilization could be judged by the way animals were treated. And no, I just wanted to finish on that because we always humans, we always put ourselves at top of the food chain or top of the evolution, and we just need to be back in a normal context of ecosystem and wildlife, and we live within it. They don't live around us. Yeah, but thank you.